Hey, did you know that with the help of a little bacterial immune system, E. coli can grow even in some pretty deadly conditions? In this video, I'll show you how I found this out. So, my name's Abby, and for the past couple of months, I've been learning some super cool things about genes. And with genes comes the even cooler world of gene editing. Gene editing allows us to make edits to an organism's DNA to change certain proteins or amino acids that allows the organism to react in totally different ways than before. So to learn a little bit more about it, I decided to conduct my own experiment. I had 3 goals with this experiment. First was to get hands-on experience editing a gene. Second was to learn about a specific case of gene editing. And lastly was to successfully complete the experiment and grow E. coli on strep plates. For a couple of my goals, I had to enlist the help of a website that has a bunch of different CRISPR kits for scientists like me who don't always have access to inventories full of streptomycin and E. coli lying around. I used this kit right here, which has all the tools that you would need to do the experiment for yourself. So before actually doing any mad sciencey stuff, there were some things that I had to set up. This would be the place that I was going to work, and then also a mini fridge. This wasn't exactly a necessary step, but just something that would make the experience a little easier. So today was just another day for me to get some extra things from the kit set up that I would need later on. So for today, this would be our LB agar plates. But what exactly is LB agar? So LB, or Lurie broth, is a medium that contains all the nutrients that E. coli would need to grow. This is basically its food supply for when it's growing on the plates. And then agar is a sort of vegetarian gelatin that's made from red algae. When it solidifies, it gives the E. coli a proper place to grow. So to set up our LB plates, we take a tube of our LB powder and 150 microliters of water and add that to a glass bottle. Once we make sure that the cap is screwed on tightly, we can shake that around vigorously until the two are basically mixed together. To make sure that the LB powder is properly dissolved into the water, we take it to the microwave to heat it up. When we're microwaving it, we need to make sure that we keep it around 30-second intervals at about normal microwave power. I didn't know what this meant at first, so I had to do a little bit of researching and experimenting, but this means that your microwave should probably be around a level 4 to 5 power. Another tip to remember for when you're microwaving your powder is to keep your eye on it. If it gets too hot, it'll boil over, and this makes a big mess for you to clean, which is not fun. Trust me, I know. You'll know it's ready when you can see through the mixture or it's basically translucent. At this point, leave it on the side for about 30 minutes or until you can handle it without too much discomfort. From that point, we're going to take 2 of our Petri plates. The Petri plates are the 7 circular plastic molds. You're going to pour the mixture into 2 of them, and then you're going to leave those for about 30 minutes so that it basically hardens. Then when it's finished, you're going to turn them upside down to avoid any condensation and leave it in the fridge. You can leave this for up to 2 to 3 months, but obviously I don't want to have to wait that long until my experiment, so I kept them in the fridge overnight. So day 2 was a little bit busier of a day because we had 2 things to do. The first thing to do was to set up our strep plates. So what's the difference between our strep plates and our regular LB agar media plates? Strep plates contain streptomycin. Streptomycin is an antibacterial that's usually used in medicines to combat bacteria because they're antibacterial. For our experiment, this is what we're going to use to test if our E. coli was properly altered or edited. While the strep plates were in the fridge, I moved on to growing the E. coli. So what exactly is E. coli? E. coli is a unicellular bacteria, and since it is a cell, it is a living thing. You can find it in the intestines of humans or also animals. But don't worry, most strains are usually very harmless. So in a lot of the other kits found on the Odin, and also other research papers and reviews, E. coli was the main bacteria of choice. It's pretty easy to grow and doesn't take a lot of expensive resources. And another thing is that we already know a lot about it. This means we don't have to deal with a lot of unknown variables when we're trying to grow it. The E. coli came freeze-dried to allow it to stay alive for longer. So to reverse the process, we added around 4 drops of sterile water to our tube, and we shook that around for around a minute. Then we micropipetted all of the bacteria onto our LB agar plates. Make sure that it's not the strep plates, since we haven't actually edited the E. coli yet, so it won't grow. After we transfer all the E. coli onto the Petri plate, we have to do something called streaking. This produces what we call colonies of E. coli over the surface of the plate. Make sure to streak the E. coli in a thin layer using long strokes with the inoculation loop seen in the video. We leave that at about room temperature for 1 to 2 days. So today is another pretty important day since we have some big things to look at and some big things to do. The first thing we have to do is check to see if our E. coli grew. So we actually successfully grew something. But wait, there's still some things that we have to do. The first thing we have to do is add our transformation mix. E. coli and DNA are both negatively charged. You think that this would bring them together, but it actually separates them, since two similar charges actually repel each other instead of making them come together. So, in order to fix this, we add our transformation mix, which actually has calcium chloride. Calcium chloride has a neutral charge. The positive ions and the negative ions are in balance, and overall they cancel each other out. So the positive ions, in this case the calcium, binds to the DNA and also to the E. coli, and the positive charges there cancel out with the negative charges. In this case, the two neutral charges will actually also bind together. If you're not totally ready to complete the next step, you can keep this mixture in the fridge at 4 degrees Celsius for about 1 to 2 days. But for us, let's move on to the next step. For the next step, we're going to be taking our plasmid and inserting it into the E. coli. The plasmid is a circular round that has a special part of it called transfer DNA, or tDNA. This tDNA is what we're actually going to be inserting into our E. coli. But what about that bacterial immune system you were talking about before? I almost forgot. CRISPR is an immune system found in some bacteria that allows it to track any viruses it's been infected with before. When it's partnered with another protein system called Cas9, it's able to remember the DNA templates of the virus, track and find it, and cut it before it makes any damage to the cell. After a little more research, scientists were able to find something super revolutionary. They found that the template of DNA that the system is trying to find can be changed to anything that you want it to find. For our experiment today, we're going to use the CRISPR-Cas9 system to find an amino acid called lysine. Lysine has the DNA template AAA or AAG. This DNA template can actually bind to streptomycin in the ribosome, and this stops the ribosome from making any proteins. If the cell doesn't have proteins, it can't carry out any of its tasks, and the cell will eventually die. But if we can change the lysine into another amino acid that the cell needs and won't cause harm, we might be able to stop the streptomycin from binding to the ribosome. Oh wait, 309. 309 has the DNA template ACA or ACG. It's very similar and they're both amino acids, but the DNA template is different, so streptomycin won't be able to bind to it. The DNA found in the plasmid actually has the C that we're going to need to change lysine into 309, which will stop the streptomycin from binding to the ribosome, saving the E. coli. If you'd like to get a more technical view of how CRISPR and gene editing actually work, I'll link my article in the description box below. The plasmid also came freeze-dried, so to bring it back to room temperature, we added 2 drops of sterile water and shook that for about a minute. Once it becomes liquefied, then we can add it to our competent cell mixture from before. To give our E. coli the best chance of developing and growing later on, we let this incubate in the fridge for about 30 minutes before moving on to the next step, which is heat shock. Heat shock is a process that uses a higher temperature than the E. coli is used to, around 37 degrees, in order to create holes in the membrane or the cell's walls. This allows the plasmid, which contains the DNA, to slip through the holes and have a better chance of actually being received into the E. coli cells. Here we're going to take a cup of 37-degree water. 37 degrees is warm enough for your hand to tell that there's been a change in temperature, but not too hot that you burn yourself in the process. We're going to incubate this mixture with the plasmid and the E. coli for 30 seconds in the water. Remember the luria broth I was telling you about earlier on in the video? This time we're going to use the same powder without the agar to give our E. coli something to feast on while it's on the plates. For this step, we're going to take a smaller tube that has just the LB powder and add water until it's almost full, but not exactly. This leaves room for shaking later on. Once we shake the tube and the LB powder is basically dissolved, we can take half of the medium and put it into our mixture with the E. coli and the plasmid. Now we should have a tube with 3 things. First should be the E. coli, then the plasmid, and third is the LB media. Incubate this mixture at 30 degrees for about 1 to 4 hours, or to give it the most time possible, I incubated it at room temperature for up to 12 hours. To properly add our E. coli to the strep plates, we're going to take 4 to 5 drops of our transform mixture and streak them onto our strep plates. And now, we wait. If you're incubating your strep plates at 30 degrees Celsius, you'll only need to do this for 24 to 36 hours. But if you're leaving yours at room temperature like I am, this is going to take a little longer, from 36 to 48 hours. 48 hours is finally up. Let's go check. So, did we succeed? Let's check our goals. Did we get a hands-on experiment of gene editing? I would say so. Did we learn about a specific case of gene editing? Yeah. Did we allow E. coli to grow on strep plates with streptomycin? We did. But there are some things that I would fix about the way I did this experiment. First of all, I would spend more time learning how to streak the plates properly. As you saw in the pictures, the E. coli grew pretty thick on the LB agar plates and also on the strep plates. But I still had an awesome experience attaining my goals. Thanks for coming along the ride, and I hope you learned something new. Bye!